Yeah, sure.
Sir, father video.
Hello.
Sir, CBS sir, principal father video இதுல இல்ல.
இல்ல ஃபாதர் ஸ்விட்ச் ஆஃப் பண்ணிருக்காரு. இது வீடியோ
இல்ல ஓபன் பண்ணிட்டேன்.
எஸ்
ஓபன் பண்ணிட்டேன்.
இப்பதான் ஆன் ஆயிருக்கு.
ஸ்விட்ச் ஆன் பண்ணிட்டேன்.
ஓகே.
Am I seen? Yeah.
Shall we start, Father?
Yeah. I'm ready.
Yes.
Yes, brother. I'm ready.
Yes. Okay. Yes, we can start, moderator. 20.
A very glorious dawn to one and all present here. At this moment of pleasure, I feel highly privileged to host this national webinar on human origin, health and diseases conducted by the Department of Botany, St. Joseph's College Autonomous, Trichy. Outwitting every hurdle in its way through Ignatian principles for more than 175 years, the first heritage institute of India to receive first A++ NAC accreditation in Tamil Nadu, St. Joseph's College has the never-ending spirit with its systemic management, allied departments, experienced staff, and dedicated students. So on behalf of the Department of Botany, I thank the management of the college for giving this 107 years old traditional department a golden opportunity to conduct this national seminar. The Center for Cellular and Molecular Biology, CCMB, is a premier research institute in frontier areas of modern biology, which conducts high basic and quality research training in frontier areas of modern biology and promotes centralized national facilities for new and modern techniques in the interdisciplinary areas of biology. From such a premier institute, it is our prerogative to have an eminent personality from CCMB, J.C. Bose Fellow and Chief Scientist Dr. K. Tangaraj, as our resource person of the day to give us the best view of the human origin, health and diseases, which has come into play more powerful in recent times. This need of the heart topic will definitely give us the deep understanding on the mechanism of body systems in relation to the external environment and our evolutionary benefits of being humans. With this brief introduction, let us get into the national webinar by invoking God's blessings with a fervent prayer song.
You are my strength when I am weak. You are the
At this blessed juncture, I now invite our beloved head of the department, Dr. S.R. Sindhil Kumar, to welcome this e-gathering. Please, sir.
Greetings in His holy name. In a paper published in June 4, 2012, the Proceedings of the National Academy of Sciences, an international team of researchers led by scientists at the University of California, suggested that inactivation of two specific genes related to the immune system may have conferred selected ancestors of modern humans with improved protection from some pathogenic bacterial strains. Now the research is moving this direction interestingly. Very good morning to all of you. The Department of Botany is organized a notable webinar lecture today on human origin, health and diseases, provides an opportunity to understand the topic and interact with the subject expert from the CSR laboratories of our country. Here, Reverend Father Dr. Araksami Xavier S.J., our beloved principal of this great institution, is known for his determination. Our college achieves greater heights in the recent years. I am sure, Father, under your leadership and guidance, may we reach next level of glory in higher education in the country. On behalf of the Botany Department, I bid a hearty welcome you, Father. Dr. K. Thangaraj, Chief Scientist, CCMB Hyderabad, who is the speaker of today's webinar, is here. His expertise in the field of origin of modern human and mitochondrial DNA variations with several disorders. He will be introduced later by Dr. Francis Xavier in detail. On behalf of the college management and the botany department, I extend my hearty welcome to the speaker, scientist Dr. Thangaraj. Special note of welcome to Dr. Sahaj Sadish, Dean School of Biological Sciences, the organizers of the webinar, Dr. Francis Xavier and Dr. David Raja, our department colleagues, and Dr. Kanikiraj in a special way. Finally, I welcome all the webinar participants of various institutions from different parts of our country and abroad. I once again welcome one and all. Thank you.
Thank you, sir, for your words of warm welcoming. With these positive vibes, I now call upon the man behind every laurel that has enhanced the quality of education and college life vibrant in our institution, our head principal, Reverend Father Dr. M. Aragasamy Xavier S.J., to felicitate the gathering with his encouraging words. Welcome, Father.
Thank you. Distinguished Chief Guest for this webinar, Dr. T. Thangaraj, CSIR, CCMB, Hyderabad. Dear and beloved Dean, Dr. Satish, dear and beloved HOD, Dr. Sendil, the organizing secretaries of this webinar, Dr. T. Francis Xavier, and Dr. H. David Raja, Dr. Kanikiraj, and dear and beloved participants, good morning to you all. I am very happy to be associated with you all, dear guests and faculty members and participants during this national webinar on human origin, health and diseases. As you all know, the lockdown period has brought us more academic exercises, particularly the conducting webinars. I am very happy that Botany Department is continuously organizing webinars, e-conferences, and whatnot. I am very proud that the department is wonderfully engaging itself in such not only academic exercises, but also some research pursuits. So first of all, I would like to appreciate and congratulate the department, particularly the organizing secretaries, Dr. T. Francis Xavier and H. David Raja, for organizing this useful, wonderful national webinar on human origin, health and diseases. My dear participants, as you all know, it is very, very important to know the human origin. Of course, along with that, our health and diseases which are affecting us. This will be very useful seminar as the distinguished resource person is from that very same field he will surely enlighten us all with his expertise, as Dr. Sindel said in his introductory remarks, welcome speech. Surely he will enlighten us, and also he will enkindle us, and also he will invite us to engage in such useful academic pursuits. And this webinar surely will bring all of us together in this academic and intellectual exercises. So I wish you all the best. I'm sure this will enlighten us a lot. May God bless you. And once again, I congratulate and appreciate the department, particularly the organizing secretaries. I also would like to sincerely thank the chief scientist of CSIR, CCMP Hyderabad, Dr. Tankaraj, for his availability, for his readiness to share his expertise with us. Thank you, Professor. Thank you, professors. I wish you all the best. Have a wonderful day. May God bless all of us. Thank you.
Thank you, Father. I now invite the person behind this webinar, the person who has put greater efforts for bringing us together for this Need of the Hour webinar, the organizing secretary, Dr. T. Francis Xavier, Assistant Professor of Botany, St. Joseph's College, to introduce the resource person of the day, Dr. K. Tangaraj. Welcome, sir.
Respected Reverend Father Principal, respected resource person of the day, the Dean, School of Biological Sciences, HOD of Botany, Dr. David Raja, one of the organizing secretary of this national webinar, and the faculty members of our department, and my dear participants. Good morning all. At the outset, I thank and welcome our resource person. He kindly accepted our invitation. Many of you know he is a very busiest scientist in CSR institution. Though he is very busy in his research activities, he kindly accepted our invitation and come over here for this national webinar. So it is my bounden duty and a pleasant duty to welcome and introduce our resource person, Dr. K. Tangaraj. He is a chief scientist and a group leader at the Center for Cellular and Molecular Biology, CCMB, Hyderabad, CSR Institute, Government of India. His main researches are origin of modern human, cardiovascular diseases, mitochondrial disorders, and male infertility. He has published over 250 research articles in well-reputed journals with high impact factors, such as the topmost journal in the scientific community is Nature. So Sir has published more than 5 research articles in Nature. He is an elected fellow of Indian Academy of Sciences, Indian National Science Academy, and the National Academy of Sciences. He is a recipient of a prestigious J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Science Award, and many more awards he received. He is a board editor in Mitochondria, associate editor of the journal BMC Medical Genetics, BMC Genetics, Tropical Medicine and International Health, and he is a member of editorial board of various journals such as Scientific Reports, Human Genetics, and Clinical Genetics. He was a president of Indian Society of Human Genetics from the year 2011 to 15, and he is a founder of the Society for Mitochondrial Research and Medicine. Such an eminent scientist and resourceful resource person with us, and a good human being with us. So though he is very busy with his academic activities, when we approach any time, he kindly accepted our invitation. So with this, I wholeheartedly welcome on my own behalf and the St. Joseph's College management behalf and the participants behalf, I welcome you, sir.
Thank you. Can I continue?
Yes,
Thank
sir.
you, Dr. Xavier, for kind introduction. Reverend Father Avikaswamy, the principal of St. Joseph's College, Dean, head of the departments, and the organizers, Dr. Xavier and Dr. David, and all the faculty members who are attending this webinar, and all the participants. A very good morning to all of you. It's my great pleasure to talk to you about the human origin, health, and diseases. So we've been working on this field for last 2.5 decades, and it's my pleasure to share all my findings. So with that, I'll go ahead and share my slides. Is it visible and am I audible?
Yes, sir. Yes, sir. Very clear.
Okay,
Very
great.
clear, sir. Nice.
So by looking at this topic, human origin, health, and disease, probably may be wondering that what is the connection between our origin and health and diseases. To give you some information about this, if you look at the way one of the evolutionary biologists, Dybzhansky, he has written an article in a journal called the American Biology of a Teacher, where he has mentioned that nothing in biology makes sense except in the light of evolution. So I'm going to take to you that how understanding our origin helps in best understanding of our health and diseases. So you all must have read about Darwin and his findings. He wrote a book called The Origin of Species, where he has explained about origin of almost all the organisms, right from orchid to whales. But what was missing in that book was that how human might have originated. Another evolutionary biologist, Thomas Henry Huxley, in his book, which is written in 1863, it's called as Evidence as to Man's Place in Nature, where he was arguing that probably we are descended from apes. But later in 1871, Darwin wrote another book called The Descent of Man, where he declared that the chimpanzee and gorilla are our close relatives based on the anatomical similarities. But now, with the availability of whole genome sequencing of human, chimpanzee, and many other organisms, it's very clear that human and chimpanzee have diverged from a common ancestor called hominids about 6 to 7 million years ago. So during the last 6 to 7 million years, how our anatomy and various aspects have changed from chimpanzee-like phenotype to human like what we look like today had happened. There are several changes. One of the changes is that we have ability to walk with 2 legs. It's called as bipedalism. Another ability is to making tools for gaining food and so on. So in Homo habilis, it started with primitive stone tools, and slowly we gain to make modern tools for killing animal and gathering food and so on. But another very interesting feature is the increase of brain size. If you look at in the early species, chimpanzees, the brain size was as similar as chimpanzee. But it is slowly increased in Australopithecus. The brain size was 35% of the modern human. In Homo habilis, 50%. In Homo ergaster, 60%. And slowly increased. And it was predicted that it was even bigger than the modern human in Homo neanderthals. Okay, this species is no more, but there are several theories why it is not there today. One of the interesting hypotheses given by Paul Mullers from Cambridge, was that the inherent violent behavior of modern human probably responsible for vanishing of this particular species. We don't know, probably might be true. So from there we evolved. But there are several research in the field of human origin and try to understand about Neanderthal. People have suggested that Neanderthal must have interbred with the modern human. But one of our study where we have shown, where we have sequenced large number of population all over the world, wherever there's a dot, that means these are the place in collaboration with many scientists all over the world. We have sequenced the whole genome of 300 individuals and try to find out about how much of Neanderthal genome is present in the modern human. It's interesting to see that except Africans, all out of Africa humans have zero, at least 1% to 3% of the Neanderthal genome. That means, so in the early days, Neanderthal probably looks like human, and it has a more brain size, must be more intelligent, we don't know. And the modern human and Neanderthal have interbred, and we have some proportion of Neanderthal genome. So coming to origin of modern human, it was very clear now that based on several research that the modern human must have originated in Africa about 160 to 200,000 years back. That was supported by a modern, the very recent study in October 2019, where they have shown that the modern human, by looking at the bone samples, it was predicted must have originated in southern part of Africa. From southern part of Africa, why did they go out of Africa and spread all over the world? This is very interesting field. Not only geneticists, archaeologists, anthropologists, historian, linguists, and also geologists have some contribution to this field. This is a study which came some time ago, 2007, mostly by geologists. So they try to understand what is the water level in early days in Africa. They found that about 135 to 75,000 years ago, there was a mega drought. The water level has reduced by 95% in a lake called Malawi in Africa. Probably that might have been the reason why the people moved out of Africa, because there's no water, no food. They are probably in a bad situation. In hunting for food, they went out of Africa. Okay, so this is one aspect. Now the question is, how did they go out of Africa? Because there are several routes to migrate out of Africa. Again, this was a very major curiosity for those who are working on archaeology, anthropology, including geneticists, to understand what was the first wave of migration went out of Africa. There are a few hypotheses. One of the hypotheses says that the first modern human might have taken the northern land route, because there's a land connection. Probably the modern human might have migrated through the land. But others didn't believe on it because the climatic condition in Europe, a northern part of Asia, might not have favored this migration, because the climatic condition even today is very cold, and think of several thousands of years back how they would have migrated towards north. On the other hand, other group of people who are working on archaeology, anthropology, genetics, and language, they believe that the first out of Africa human might have taken the southern coastal route. So what is the reason for believing on this? There are several archaeological evidence present in some other part of the world. For example, the archaeological findings in Australia suggested that the modern human might have arrived Australia about 50,000 years back. China about 39,000 years back. Middle East 47,000 years back, and Europe about 40,000 years back. Unfortunately, we don't have any good archaeological tools to suggest that when did the modern human must have arrived in Indian subcontinent. Only in the recent past, there are some archaeological evidence, but still we are not very conclusive about talking about when did the modern human arrived in Indian subcontinent. There are some evidence which suggests that one of the evidence is based on the archaeological excavation done in a place called Jolapuram in Andhra Pradesh. The group of scientists have excavated this place and found a lot of stone tools, which probably they thought that the modern human might have used these tools. But unfortunately, there was no biological remains of modern human was found in this particular excavation site. So there is no evidence that this tool might have been used by the modern human. Very recently, you all must have read in the newspaper, there are a couple of sites in Tamil Nadu. One is Kiredi, where a lot of work has been done. Until very recently, there was no bone sample was found there. Only, I think about a week back, this skeleton of a child, a couple of children have been found. And that we are planning to analyze. In fact, the state archaeological department commissioner is in touch with us, with me and Madurai Kamarajar University to analyze this bone sample to find out about the origin, and it might give you valuable information, which I will tell you much later that why it's very, very important to analyze this genome for understanding our origin. This is one side. So another site is called as Adityanallur, again in Tamil Nadu, where the Archaeological Survey of India, so that the previous one is under state government. This is the Indian Archaeological Survey of India, where they have excavated the site and collected a lot of bone samples and kept it in their office, which is within the complex of Tamil Nadu State Assembly. And recently, in the month of February, I went there, met and collected some sample and signed MOU, and we were exchanging MOU with Archaeological Survey of India. I think these two bone samples will give you some valuable information. On the other hand, where we have some very interesting finding using the ancient bones, one is from a place called Rakigiri. It's one of the Harappan site, and you know that is a place where predicted that the modern civilization must have taken place. Fortunately, we found the intact bone sample of a lady, and we analyzed that sample, and we found that this is the sample which obtained from Rakigiri or Indus Valley civilization sample, where we found it's a combination of the very ancestral population of India, the Andaman tribes. I'll come a bit later about origin of that to connect how it's important. And the large proportion is from the Iranian hunter-gatherer. Okay, why it's very important? Although the sample is not very old, this is about 3000 BCE, but we don't have a clue about when did the first modern human arrive. Okay, but this is very important finding that this sample as a combination of both the ancestral South Indians or Indian population and the Iranian hunter-gatherer. But this sample doesn't have a proportion of genome from Iranian either farmers or herders. On the other hand, if you come little out of the human origin, try to understand about when the agriculture started in India, it was believed that there's a place called Fertile Crescent in Iran. That's where the agriculture started, and probably that got spread to the rest of the world. But this study suggests that they don't have Iranian farmers' genome, That actually led us to conclude that probably the agriculture must have started parallelly or independently in India. Okay, so here that's a major conclusion, but still we don't have clue about when did the first modern human arrived or whether this sample is related to early human migration. In another study where there's a large number of scientists involved all over the world, and David is the main author, and we are some of the major senior authors in this. But this study mostly on the ancient sample. There are about 500 and plus bone samples obtained from Middle East and Central Asia, where we have analyzed this genome and concluded that probably the place where in the Central Asia, there's a population called Yamuna steppe pastoralists. So they are the one must have source population for Indo-European languages or Indo-Aryan languages. So we are not talking anything about that. But what was interesting is that they have migrated towards north and come to India. It's only about 1000 to 2000 BCE, not very old. Again, recent times in terms of the early human migration. So there's no combination of no proportion of that Yamuna steppe pastoralist in South Indian population, only in North India. On the other hand, if you broadly look at, they're the people who also taken the Indo-European language to the Europe. Okay, so this study also is not talking about the early human migration. Okay, so although we don't have a good archaeological evidence to analyze and predict that, okay, this is the first modern human who migrated several thousands of years back. However, we have a large number of population groups in India who maintain their endogamy, and we thought that analyzing their genome would give us valuable information. As you see, India as a whole, and it's not exaggeration to say that India is an ethnic museum because we have collection of large number of population groups. So these groups are stratified in many ways. That's one of the interesting aspects of the Indian population. Broadly, they have been classified based on the language. As you know, there are 4 major linguistic groups: Dravidians, Austroasiatic, Tibeto-Burman, and Indo-European. These are the 4 major linguistic groups. Again, under each language group, there are several languages. For example, if you take Dravidian language, Tamil, Malayalam, Kannada, and Telugu, these are the 4 major languages under Dravidian linguistic groups. Similarly, Austroasiatic have several languages, Tibeto-Burman has several languages, and Indo-European has several languages. In addition to these major language groups, there are several isolated groups like the Andamanis, Nihalis, and so on. Another interesting aspect is that under each linguistic group, there are several social groups. As you know, the caste system, tribal, again, the caste, we have upper caste, middle caste, lower caste, tribes, primitive tribes, hunter-gatherer, and so on. So putting all of these makes India a very complex country in terms of our social structure. You may be surprised to know that in India we have 4,635 anthropologically well-defined population groups, including several tribes, primitive tribes, and 36 of them still follow hunter and gatherers lifestyle. That means they just hunt for the food. They don't do any job, including agriculture. They go and hunt for food, eat, sleep. Again, next day they'll go and hunt for the food. So as we know that these are the number of population groups we have in Indian subcontinent. So now, based on this, what I'm going to talk to you is that how this population in India have originated, this many number of population, and what is the genetic affinities between these 2 groups. Okay, that's the first aspect I'm going to discuss. After discussing that, I'm going to talk about how understanding the origin of our population will help in better understanding of our health and disease. First, let us look at origin of our population. We have been working on this field for last 2.5 decades, as I mentioned earlier, and to our surprise, we came to know that the first out of Africa human, this is a major question, how did the modern human migrated out of Africa and who are these populations? And to our surprise, we came to know the very first out of Africa human is living in Indian subcontinent. Who are they? They are none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware that there's a population like this exists in Indian subcontinent. They are hunter-gatherers, as I mentioned. And if you closely look at the Andaman and Nicobar Islands, there are 572 small islands, and there are 6 tribal populations, indigenous tribal groups. 4 of them are they typically look like Africans, as you've seen in the previous slides: Jarwas, Great Andamanis, Sentinelese, and Onge. 2 groups, Nicobaris and Champagne, typically look like Southeast Asian groups like Chinese, Malays, and so on. And to give you some idea about this island, as I mentioned, it's a small islands, very beautiful, very clear, as you see these waters, and it's very best place to go and spend some time. I visit almost every year because there's one ICMR Institute, Regional Medical Research Center. I'm the scientific advisory committee member, so that gives me opportunity visit every year. As in this island, very selected island, as I mentioned earlier, these tribal population have settled. But you see that numbers are very, very small. Great Andaman is only 20. Earlier, there are many groups, only Jeru is existing now. Onge 98, Jarawa 200, and Sentinel is 250. So these 3 groups, we could go near them, collect some sample. But Sentinelese, nobody could go near them. If somebody goes there, they will shoot with the arrow. I think a couple of years back, you might have read in the news that one American tourist tried to visit there and they have killed that person. Okay, so only Sentinelese are inaccessible, whereas other population we could collect sample and pursued our research. On the other hand, the Nicobaris are the one which is large in number, 20,000, whereas Champagne also small in number. One might ask the question, why they are very small in number? There could be many reasons. One reason could be they are highly endogamous population because of the high inbreeding. So the population size has reduced. But there's one insight which probably not many of them know, is that when Britishers captured India and wanted to put all our freedom fighters in a jail, so they constructed a jail called cellular jail. There are 7 wings, and 2 have I think demolished, and 5 are existing now. And this is a real view. That's a Google view. So what you see is these are the 2 wings. Okay, and if you imagine that how our freedom fighters have been kept in this, so each one this is a one cell. Each one is the window of one cell, and this is a front side. Again, each one. Now, if you go inside, you can see the cell. So if the freedom fighters, somebody is kept in a cell, wanted to see outside world, they cannot see. In the front, they can see the back side of this wing. On the back side, they wanted to see, they cannot see it because it's covered. Okay, that's how our freedom fighters have been kept. So why I am bringing this is that for constructing this cellular jail, they have killed large number of the tribal groups. That could be one of the reasons why the number is reduced. After killing, one good thing what they have done was they have taken the skull and hair samples and kept it in the Cambridge Museum Duckworth Laboratory Museum in Cambridge. And in fact, that allowed us to collect some sample from these bones, analyzing them and compare with the modern or living Andamanese. Okay, at least they could preserve these bone and hair samples. So what we did is we went to these islands. This is Onge. They are living in an island called Dugong Creek. It's very difficult to reach there. In fact, so I explained that information in somebody interviewed me in Time magazine. Of course, that is kept here, so I'll not go into detail. But we collected this sample of Onge, some samples from Jarva, some sample from Great Andamanis. And we have done several work. And the first thing anybody wanted to do the evolutionary study or try to understand the population history, we use the mitochondrial DNA. As a biology students all know that in a cell, there's a nucleus and there's a large number of mitochondria in the cytoplasm, and each mitochondria has large number of mitochondrial DNA. The reason why we use the mitochondrial DNA is that all the mitochondria we all inherit only from the mother. There's no paternal contribution of mitochondria or mitochondrial DNA. That's one aspect. The second aspect is it transmits maternally from mother to son and daughter. Son cannot transmit that. Only the daughter can transmit to the next generation without any change. Whereas the nuclear genome undergo recombination during the meiosis, whereas the mitochondrial DNA will not undergo any recombination except the naturally occurring mutation. It goes generation after generation without any change. That's another aspect. The third aspect is particularly for ancient DNA study, we have only 2 copies of the nuclear genome. One copy we have 46 chromosome, 23 we inherit from father, 23 from the mother. Right? Whereas the mitochondria, they are large in number, hundreds, sometimes thousands, because this is two copies, as I said. There's a large number of mitochondria, and each one has a large number of mitochondrial DNA. There are hundreds of mitochondria in a given cell. Therefore, the copy number is more, and they are circular in size. Therefore, that doesn't undergo kind of degradation. So most often they are intact compared to the nuclear genome. So these are the properties why we use mitochondrial DNA for any evolutionary studies. So what we did, we sequenced the complete mitochondrial DNA of the Andamanis, 16.5 kb. When you sequence, those who are not familiar with this, so this is the sequence of 2 different individuals, and there are 4 colors representing 4 different nucleotides. The blue one is C, green is A, red is T, and orange is G. So these are the 4 colors, and depending upon the sequence, you'll see different color pattern. This is a sequence of one individual, and this is a sequence of another individual. As you see, there are some places where you see the dots. That means this is a place there's a difference between these 2 individuals. You can see clearly that this sequence has G in this position, whereas this sequence has A in this position. Same way, this sequence has A in this position, this sequence has G in this position. So when you sequence a complete mitochondrial DNA, you might find a lot more variations, particularly in the non-coding region. Some are highly polymorphic, some which are in the coding region might lead to a large number of diseases, particularly the neuromuscular disease. That's another big field. I'll not talk about that now. Again, in the polymorphic region, some are evolutionarily highly conserved. Those are the ones which we use for constructing the haplogroup tree. So this is a worldwide mitochondrial haplogroup tree, and you see that this is a parsimonious tree. This is defined by set of mutation in the mitochondrial DNA. The L-derived haplogroup L1, L2, and L3 is the root of mitochondrial tree that you will see only in Africa, as you see here, with a high proportion. But you can also see that the population which is migrated very recently. For example, in India, we have a population called Siddhis who have migrated recently. Migrated in the sense, who have been brought by Portuguese traders and sold them to the Nawab and Sultans to work in their army. If you analyze them, if they are pure unmixed, then you can see them. So in fact, we have seen L-derived haplogroup in some of the Siddhi population, whereas you don't see that in any other population around the world. And similarly, each country or continent, you can see very distinct haplogroup distribution. For example, in India, you see large number of Hem haplogroup that is immediately after L. With this mutation, there is additional set of mutation defines the Hem haplogroup. If these mutations are not there, additional mutations will put them into N. Then subsequent mutations help in putting in different haplogroup. As you see all over the world, different population or different countries, continent have different haplogroups. When we sequence the mitochondrial DNA of the Andaman tribes, and we expected probably they may fall into L-derived haplogroup because they are phenotypically similar to Africans. But to our surprise, they did not fall into this haplogroup. Rather, we have found large number of mutations. These are the mutation site. As you see, there's a large number of mutation which is present only in Andamanis, but not in any group outside Andaman, okay, including in India. So these are the unique set of mutation, although phenotypically they are similar with Africans, but their mutation is totally different. Okay, so based on this, one can also estimate the age of this population, and based on that, we assign a new haplogroup called M31 and M32, and we propose very first time that probably the Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route and reaching Andaman and Nicobar Islands about 65,000 to 70,000 years back. So this is the first genetic evidence suggesting the first modern human who migrated out of Africa has taken the southern coastal route, and they are none other than tribal populations of Andaman and Nicobar Islands. So this is the very strong evidence, and probably people from here, they migrated towards Australia. The Australian Aboriginals, Andamanis, and some group in southern part of India share a common mitochondrial DNA mutation, and it's very clear there's no contradiction about this migration. In the same issue of Science, 2005, there's another group who studied a population called Orang Asli from Malaysia. So they have also come to the same conclusion, and there was a perspective or commentary in the same issue by Peter Foster, citing both the paper, our own paper and Macaulay's paper, the previous paper, and shown that this is a plausible way of migration of the first out of Africa human. So this is still holding true that the first out of Africa human migrated taking southern coastal route, and they are Andaman tribes. Okay. After looking at this, after the whole genome of human sequence, and there are companies which have identified about 1 million genetic variation across the human genome and made us a chip. So that allowed us to look at several individuals' genome-wide variation. We thought looking at that will address very important issue, not only the evolution and also some aspects of our health and diseases. So now we used the genome-wide SNP markers. For that, we carefully selected 132 individuals representing 15 states. Again, we used very carefully population based on the mitochondrial DNA. We also looked at the Y chromosome marker because most of the populations which we studied are males. Based on that, we don't want to use the sample or duplicate the sample. Therefore, we clearly avoided the close related individuals and taken few representative samples from each population and each linguistic groups. Therefore, we can have a very fruitful conclusion. We used Affymetrix array 6.0, which consisting of 1 million SNPs. We used human genome diversity panels and HapMap dataset. By comparing all these, we eliminated all the marker which are not useful and end up in 560,000 SNPs, and we did a lot of analysis. So one of the analysis is called PCA, that's a principal component analysis. See here we have a lot of Indian groups, we have European HapMap group, and this is a European African HapMap group, and this is a Chinese HapMap. Now you see that all these HapMap samples are falling in a single cluster, suggesting that genetically they are more homogeneous, not variations within their genome, therefore they are in single cluster. Whereas, look at the Indians. Each Indian population is very unique, therefore they clustered uniquely. Again, another interesting aspect is that although each population is very unique, but still they have genetic affinities with other groups. This kind of gradient, what you see, suggesting that each population have some genetic affinities with other populations. On one hand, you see the Indo-European group, Kashmir Pandit, and other group, you can see the Great Andamans. It's the gradient of a relationship that suggested that India is not inhabited by a single large population, as you see here. Rather, we conclude that India is inhabited by large number of small population groups. So what is this genetic affinities, which I will come with later. So what was the conclusion based on this? Was that in the prehistoric India, there were two founding populations. One, we call them as ancestral South Indians. Probably when Andamanis were migrating out of Africa, this is a deep-rooted divergence, and some group must have settled in southern part of India, whom we call as ancestral South India. Okay, you may ask that why nobody is typically like either Africans or Andamanis, because we have admixed. For that point, I'll come again later, that because of the admixture, the original feature has gone. But you may see still there are some groups who are with the dark skin color and maybe with curly hair and so on. But very rarely we see the typical phenotype as the Andamanis. So this is the first modern human probably settled in Indian subcontinent. The Andamanis, after that, before going into Andaman, some groups settled in southern part of India. And as I mentioned, that why looking at the Curedy and Achanalur samples are very important, because that might give you some clue about that early human migration. Then the second population which settled was whom we call as ancestral North Indians. There was second wave of migration. People migrate towards north, and we believe that some group has gone to Europe, and one group has come to northern part of India through Middle East and settled in northern part of India. But it's not very old as compared to this, but probably it's 35 to 40,000 years back. But we don't have a very clear-cut evidence about the migration of ANI. But what we know is that these two, the relationship between ANI or ancestral North Indian and Europe, because there are some mutations which are found in Europe. For example, the skin color mutation, because everybody is originated from Africa. Why some people are lighter in color? Because that's because of the evolutionary pressure. There was a mutation in the people who are living in very cold country where they don't see sunlight, because sunlight is essential for vitamin D synthesis. where the dark skin color might not have helped in penetrating the light. Therefore, the melanin content has to be reduced. Where we found that this gene called SLC24A has a mutation which made the skin color turning from dark to light color. The same mutation is also present in people in the Middle East, in the northern part of India, but not in southern part of India. And one more evidence is the Y chromosome haplogroup, R1A, is present in Europe, Middle East, and also northern part of India. The third one is the lactate persistent gene mutation, which helped in digesting the milk sugar. Is also common between Europeans, Middle Eastern, and particularly the people living in northwestern part of India, where the agriculture and domestication of animals have taken place. So this all suggesting, again, I'm not going to detail because I have to cover most of it. So to give you evidence that why we believe that ancestral North Indians have European connection. So based on that, what we concluded is that these two groups, ancestral North Indian and ancestral South Indian, must have admixed in some point of time. We don't have evidence at that point of time when did the admixture must have took place. Because this tells that why in India we have different phenotypes, and even one particular phenotype, if you take the skin color, we have range of skin color. So these are all because of the admixture between these two groups. Okay. The conclusion at that time was all the Indians have admixed, and there could be high frequency of recessive disease because of the following endogamy marriage practice. With this evidence, now we wanted to move on to understand when this admixture must have took place between ancestral South Indian and ancestral North Indians. And we have again studied large number of population from southern part of India and large number of population from northern part of India. As I mentioned earlier, there will be a recombination during the meiosis, and when during the meiosis, both fathers and mothers genome which that individual inherited will undergo meiosis. That means the homologous chromosome will exchange the chromosomal material before it is transmitted to the next generation through the germ cell. So using that recombination, we try to estimate when this admixture must have took place. So in this study, what we have shown was this: initially the ancestral South Indian and ancestral North Indian must have given rise to many population groups individually. Okay. Then during the last 2,000 to 4,000 years, these two founding groups have admixed. As the color shows, they have different proportion of the two founding populations genome in their population. But after admixture, for the last 2,000 years, every single population started maintaining their endogamy. Endogamy means marrying within the group. Okay, that means, so whatever the changes in our genome, because naturally there will be some mutations, whatever the changes happen in every single population remains there within the population. Okay, that is what making every population as unique, because when you do the genetic analysis like a PCA, how it is differentiates population, because if there is a mutation in one population which is not present in another population, it distinguishes. So that's the reason every population is unique in terms of evolution. And what we found also that the unmixed ANI and ASI is no more. So we didn't find any other population which we analyzed are unmixed except the Andaman tribes. Okay. So using this information, so now what we wanted to do is that, as you know, to summarize this part, this is a population which migrated out of Africa, reaching Andaman and Nicobar Islands about 60 to 65,000 years back. And while migrating, some group stayed back in southern part of India, whom we call as ancestral South Indian. There was also movement much later towards north, and there's a group reached northern part of India, whom we call as ancestral North Indians. And these two groups have admixed. After admixture for last 2,000 years, they maintain the endogamy. Now, what I'm going to talk to you about is that how this endogamy is having influence on our health and disease. Okay, so that's my second aspect of the talk. To do that, this time we didn't restrict to analyze only the Indian population. We thought it would be interesting to study the entire South Asia. Because when you talk about entire South Asia, now we have 5,000 population. And there's no other country in the world or region in the world has such a huge human diversity, right? That's one aspect. The second aspect, why we wanted to go out of India, taking South Asia as a whole, there are several reasons. One of the reasons is that from our own study, where we have studied, there's a disease called cardiomyopathy, and there are several genes responsible for the disease. So one of the genes called myosin binding protein C3 genes, very, very important gene, and this connects both the protein is involved in contracting the contraction of heart muscle, which connects both thick and thin filament of the heart muscle. It's very, very essential. If there is any mutation, it will lead to cardiomyopathy. Cardiomyopathy is a heart muscle disease, and if the mutation exists in the heterozygous condition, that means in a pair of chromosome, only one chromosome carries the mutation, other chromosome is normal. The individual live very happily up to 40 or 45 years. But after that, it will lead to sudden cardiac death. But if the mutation is in the homozygous condition, and the individual die at very, very early age. How did we know that? We analyzed large number of post-mortem tissue of the children who died at very early age, less than 10 years, and we found this mutation in homozygous condition. So this is one example where the child was 3-year-old, I think. The heart shows dilated cardiomyopathy, it's enlargement of ventricle. And this other case where you see the thickening of heart muscle, hypertrophy, the intraventricular septum is so thick. This is one of the features of the cardiomyopathy. But another interesting aspect for the student is that this mutation, the 25 base pair deletion, which we found in these patients, is in the intronic region, and therefore intronic region is also very important and it play a major role in causing the disease. So we should not ignore the intronic mutation, because that skips away one of the wild type exon. Therefore, there's a truncation of the protein, and it also alters the cardiomyocyte structures. This is a wild type where you can see the cardiomyocytes are beautifully arranged, whereas in the mutant, where you see this is the animal experiment, you can see this array of this mutant. Okay. So now I am coming back to the topic. So this is the basic that why we wanted to include the South Asia. When we try to see the distribution of this mutation all over the world, to our surprise, we found that mutation is only present in Indian subcontinent or South Asia, but not in other countries. The one which is in yellow are the one which carry the mutation in approximately 4% in every population in the South Asia. So that suggests that whatever we find in India, it's also true for other South Asian countries. Of course, we found that this mutation originated in about 30,000 years back, because when it's heterozygous, it's not going to cause any disease up to the age of 40 or 45 years. By then, the individual get marries and pass on to the next generation. That's how it got well spread. Now, how it is important, how it is distributed all over India or South Asia is a question. Because we know that there are two founding groups, these two have admixed the last 2,000 to 4,000 years, and this mutation is 30,000 year old and originated somewhere in India. Then because of the admixture, it got well spread across Indian subcontinent. So because that helps us in understanding our evolutionary biology, helps us in understanding the spread of this particular mutation. So that's the reason we wanted to now see the entire South Asia rather than only looking at Indian population. Now we took 2,800 individuals from 275 endogamous group. All this, as you see that this color indicate, these are the linguistic affiliation of these samples. The sample includes, as I mentioned, all the South Asian countries, 1 million SNP. We try to understand many aspects. So one of the aspects which is very interesting is that most of the population which we analyze, at least 1/3 of the Indian populations are South Asians, have high IBD score. IBD score is the identity by descent. Technically, if you want to tell what is that, if a large stretch of DNA strand from a common founder in the last 100 generation. If you wanted to say in simple word, if you take two unrelated individuals, you know that if you compare two unrelated individuals, we expect to see variation between these two individuals at every 500 base or 1 kb region, right? If they are unrelated. But they may say that they are unrelated, but if they are come from a common founder, we may not see any variation. So if you look at the stretch of DNA fragment, or we can otherwise call as haplotype, there will not be any difference if they have come from a common founder. For example, go to some remote village, you might find there are some 50 families or 100 families, and all the 50 or 100 family must have come from one single founding parents. Okay, the founders might have had 4 or 5 children those days, right? So, and each one married, again, they have 4 or 5 children, then again 4 or 5 children. So that entire village, you see that they have come from a common founding parents about, let's say, 30 generation or 50 generation back. Okay. So the reason for that and the impact of that founding population marrying within the group has several issues related to our health and disease. As I mentioned in one of my slide, where we expect that every individual or every generation might have a naturally occurring mutation. If the mutation is in non-coding region, there's no problem. But if that is essential in the coding region, and it is going to cause different phenotype, right? And again, the mutation is dominant in nature. Even one chromosome of a pair, as then it will lead to some disease. Okay, one can see obviously, okay, this is the reason for this. But if the mutation is in recessive in nature, the recessive mutation, if it is existing in heterozygous condition, it means one of the pair has a mutation, is not going to cause any phenotypic change. Okay, the individual will be carrier and he or she will be absolutely fine. Okay. But if the mutation exists in a homozygous condition, only then it's going to cause the disease. Okay, cause a recessive disease because the recessive mutation. And in this scenario, where there's a founding parents or founding group has a large number of individuals, because of not only the consanguinity is marrying within the family. So this is what I'm talking about, marrying within the group or population. If there is a founding mutation which is recessive in nature, and that mutation is going to circulate or enhance in the population. Until otherwise, the individual carry a homozygous mutation, it's not going to cause any phenotype. Okay. Only when it happen to be in homozygous condition, only then it's going to cause the disease. So that is very, very important point for looking at the population which has very high IBD score. Look at here, each line represent one population, and these two populations which are highlighted in red color, Ashkenazi Jewish and Finnish population, they are a very small group and known for their population specific diseases, which I'll show a bit later. But you see that they are well known and they have large number of disease. Look at most of the Indian populations are having several fold high IBD score, means that there's a lot of problem, particularly the recessive mutation in large number of Indian groups, which nobody knows now, because this population has been collected for the population study. We don't know whether any disease exists in this population or not, right? So now, in a simple way, I'll tell you that as a biology, students must be aware, this is a pedigree. Circle means female, this is a male. If you put line between these two, it's a marriage between these two. And if you put line between in the middle of this down, and that means these two are their children, two daughters, and these two have married somebody from outside. Then if you consider that this is a mutation, a recessive mutation exists here, because it's heterozygous condition not present here, therefore this individual is fine. Then she passed on to the next generation. She married from somebody outside. There's no mutation. Again, fine, is going in heterozygous condition, generation after generation. Okay. So now, after few generations, they might have lost their relationship. Okay. They are in the same community, but they don't know whether they are related or not. But now, after a few generations, and you can go after a few generations also, now you see that because this happened because of the recombination during the meiosis. Now, both this boy and girl, the same community, carrying the mutation in heterozygous condition, same mutation, absolutely fine. But when they marry, there's a 25% chance that they're going to contribute these two abnormal chromosomes. Therefore, there's going to be a disease. I'll give you some example in the coming slides. Now I'll talk about what I showed here, this Ashkenazi Jewish and Finnish population, very tiny IBD score, but still they have large number of diseases. These are the number of diseases, and most of them are autosomal recessive in nature. Okay, and these are the genes. Similarly, Finnish population. Okay, now you can imagine large number of Indian population, they have several fold high IBD score, and what would be the consequences of this? That's what we are going to explore now, right? So that to understand that hypothesis, we took one well-known recessive disease called progressive pseudorheumatoid dysplasia, and the age of onset is very, very early, 3 years. And you see that it's a joint, swelling of joint. Any joint you will take, this is unfortunate that these children have to undergo a lot of pain. As you see, the joints are swollen, and the X-ray also shows that. We took some 33 patients. We sequenced and found whether this mutation is present or not in WSP3 gene. And 6 patients, the children, carry that mutation in a homozygous condition. Parents are absolutely fine because both are heterozygous. Then we wanted to see whether all the 6 have come from a common founder or not. So we sequenced the flanking region of the mutation to see the haplotype, extended haplotype, to understand whether all the 6 have come from this common founder or not. We found 5 out of 6 individuals who now claim that they are non-consanguineous and not related, but looks like they have come from a common founder. Several generations back, they are related, but now they don't know whether they are related or not. So that tells that this is the impact of the founding mutation and endogamous marriage. Okay. So to understand and give the scientific community, particularly those who are working on genetics. We publish this paper putting all this information, and particularly it's useful for the researcher and students to know that there's a lot more in Indian subcontinent to understand and discover a lot of population specific recessive disease. To explain that in a simple way, I made this diagram which the editor liked and put it on the cover, where there are a couple of information from this. So one is that in Indian subcontinent, people are living as a group, as you see here, right? But not in the other cases. And because of the endogamous marriage, marrying within the group, there could be some founding mutation, which is recessive in nature. And because of marrying within the group, the heterozygous mutation, as it's shown, I'll come closer to that. The individual with heterozygous mutations are increasing in number. And there's a chance that the boy and girl who carry the heterozygous mutation marries, and as you see, it's a full color, suggesting that these are the individuals who carry the mutation in homozygous condition. Therefore, there's a disease. On the other hand, people living in other continent or countries also have mutation in heterozygous, but because they don't follow strict endogamy marriage, and not necessarily they are going to find somebody with the same mutation, marries, and giving rise to child or children with the homozygous mutation and the disease. So having this, we try to understand that in other diseases which we are doing it. This is one of the very rare case of mitochondrial disorders, where you can clearly see the impact of the endogamy. Okay, in the first family, it's a consanguineous family, as you see here, the pedigree. How we can clearly see the impact of a recessive mutation. You see that the parents, unfortunately, we could not analyze them. The parents who are 45 years and 39 years are absolutely fine, but carrying the mutation in heterozygous condition. Okay, this is a wild type C. You can see there are two peaks, C and T, because this is a complete mutant. It's a complete mutant, wild type, and the heterozygous. Both the parents are heterozygous. Unfortunately, they contributed both of them contributed the abnormal chromosome to the next generation. Unfortunately, two children, one boy and girl, have carrying this mutation homozygous condition. While analyzing, this boy was alive, but by the time we complete the analysis, this boy was no more. But we don't know the condition of this child, and probably, unfortunately, this child also cannot be survived. And these are some of the abnormality which is led by this mutation. So this mutation is in a gene called polymerase gamma, which is essential for mitochondrial DNA replication. This single base mutation, one base mutation, and you see that this is a wild type, intact 9.9 kb DNA. So because of one base mutation in a polymer called polymerase gamma gene, and that is essential for mitochondrial DNA replication, and that error introduced a lot of deletions in the mitochondrial DNA. This is 9 kb. You can see that there's a 5 kb deletion, maybe 6 kb DNA deletion, 3 kb deletion, and therefore it introduced several mitochondrial DNA deletion. Therefore, you are seeing multiple fragments here. And these are showing the brain MRI, showing the lesions, and this is an accumulation of abnormal mitochondria called as rag blue fiber. This is a COX negative fiber. These are all some of the typical features of mitochondrial disease. In this case, this is a non-consanguineous family. Again, both the parents who are 41 years and 38 years are with absolutely fine with heterozygous mutation, and this is very unfortunate that both the children have died. Using this information, so now there's one child alive in this family, okay, heterozygous. Now this child grow and come to the stage of marriage. Actually, they should avoid marrying somebody from the same family or within the community. And even if they wanted him to marry somebody from the relative, they should actually check. Now it is very straightforward, because while sequencing, we have to sequence the whole gene, and some case we are sequencing the whole exome and the whole genome. But in this case, it takes maybe a couple of ₹100 to screen for this particular mutation and see whether the girl also has the mutation whom he is going to marry, and accordingly they can decide. Okay, so now you can also see how it's very, very important for the future generation. Okay, so with that, what I want to tell you is that knowing the information about the Indian population structure and what is the impact of the endogamy, how many population, what we see here, are carrying this, probably carrying the recessive mutation in large number, going to have some kind of fruitful thing if you do genetic based studies. What we need to do is something like what the Ashkenazi Jewish population practice. They have the organization called Dor Eshrim. What this organization does is that they go to school, they collect sample from the children, sequence them, put it in the database. Okay. When they grow old, when the boy wanted to marry a girl, and both the sequences are available in the Dor Eshrim, they will pick up the sequence of both the individual and try to see whether the recessive mutation present in the boy also present the girl. Okay, so that means they are going to, if there is a mutation, definitely they are going to have a child with homozygous condition. Therefore, that is a disease. So they advise them, give counsel that either they should not marry, or even they should marry, they should look for this particular mutation and decide their pregnancy outcome and so on. So that is the level of the genetic study, genetic counseling is happening in Ashkenazi Jewish. Having studied a large number of population and having get to know that a large number of populations are with high IPD score, it's our responsibility now to go back to the community, sequence the whole genome, and try to understand, is there any mutation? Then go back to the community and screen the community and identify the mutation, give them constant counseling. So that's our next step. So now what I'm doing is, we are taking this study in two broad way. One is where we know there are population with a specific disease. There's a couple of examples I've shown the picture. These are the population called as Handigudu. There's a place called Handigudu in Karnataka, in Shimago district. And the population, most of the individuals in that particular region are very short in stature with bone abnormality. As you see that the bone is twisted or bent, and they have to undergo a lot of pain. Okay, so this is restricted to one particular region, although there are several other studies has been done, but not complete genetic study has been done on this population. It's very, very important for us to identify the mutation. Therefore, subsequently, we can give them the counseling. That is one family. This is another example. There's a place called in a region in a coastal Andhra Pradesh, a place called Udanam, and there's a disease which are of unknown origins called as Udanam kidney disease. Okay. A large number of people have to undergo dialysis, as you see in this picture. That again, people have come up with many reasons. One of the reasons could be the water, or they do a lot of cashew nut the cultivation, and there are many more hypotheses, but nothing is coming up with very concrete evidence. Therefore, it's very important for us to understand the genetic basis of this disease, and hence it will be useful for this community. Similarly, there are a large number of diseases which are of population specific in nature. What I've shown earlier, large number of population with high IBD score, we don't know really whether there is any disease exist in that population. So this is one category where we know there's a population specific disease. On the other hand, we know that there's something is hidden in the population. Now we are sequencing both of them, a large number of groups, to understand their the population specific disease causing mutation. Therefore, it will be useful for the community in future. So with that, I conclude my talk by thanking all my colleagues within CCMB, Dr. Lalji Singh, and a lot of my colleagues and students and collaborators from several countries, including David and many more clinical collaborators who have contributed to the study. Although I summarized, each study has a large number of people involvement. With that, I also thank the funding agencies like CSIR, BK, EBG, EST, and MRC. Thank you very much to all of you for listening to this talk, and I'll be happy to address if you have any questions. Thank you very much.
Thank you, sir, for your wonderful presentation. I hope the participants would have definitely benefited from your webinar. This is now the time for Q&A session of the webinar. I request the participants to post your queries at the chat box, the comment box, such that the moderator will put forward the questions to the resource person on your behalf. I repeat, I request the participants to post your queries at the comment box, such that the moderators will put forward the questions to the resource persons on your behalf. Thank you.
Thank you, sir. I thank our resource person for his enlightened talk on human origin. So here we received many questions from participants. We selected only some, and that will be asked with the resource person. The first question is from Arpita Tiruchirappalli. So her question is, can we predict the evolution of a plant by considering the genome of chloroplast? I repeat the question. Can we predict the evolution of a plant by considering the genome of chloroplast?
Yes, there's some limitation by looking at only the chloroplast because this might be very small, like a mitochondria. But you need to use the whole genome because now the techniques are available to sequence the whole genome of a plant, any plant, which is much cheaper now. That would definitely help in understanding how many millions years back this genome or this particular species of plant evolved.
Thank you, sir. There is another question from Anash Kumar. He is from Central University of Haryana. Why is the effect of COVID-19 virus less in tribals? I repeat the question. Why is the effect of COVID-19 virus less seen in tribals?
Yeah, it's very interesting, particularly now, because there are many aspects one can discuss about it. So one is that the tribals might not have had contact with anybody having the COVID. If they are in isolation, nobody is going near them, they are perfectly fine. That's one explanation. Another explanation is that the people who are living in a very adverse environment, not like those who are living in cities, they are already exposed to a lot of pathogens, like including parasites, bacteria, and virus. They might have already got natural immunity because one example I can say that the people who are living in a very remote area in a jungle who are exposed to malaria parasites have gained a lot of mutation which is responsible for giving resistant against malaria, which I didn't discuss in my talk, but that's a different talk where we try to look at the variation exists in the genes which makes the individual susceptible or resistant against malaria, where we found the gradient. If you take population living in the city, some caste, tribes, primitive tribes, you can see the primitive tribes have high frequency of such mutation compared to tribes, compared to caste population. So that's another reason that they are exposed and naturally selected for living in adverse environment. So these are the two explanations I can give.
Yes, sir. Thank you, sir. So one more question from Swaraj Mohanty. His question is related to the high altitude and the glacier region population study of people from India. Is there any evidence of genetic stability and a mutation study on the genetic diseases? Shall I repeat this?
So there are, I think, two aspects. One is the difference between people living in low altitude and high altitude, right? So two aspects one can see: the adaptation to live in high altitude. So there are mutations which are responsible for taking high oxygen, the low oxygen level, right? Those who are living in very high altitude, the environmental oxygen may be very less, right? There are mutations which helps carrying more oxygen by RBC in those individuals compared to others. Okay, that's why they could able to survive there. So that is because of the adaptation. And because when you try to look at the disease, some of the disease where you see again the malaria example, the mutation which we see in other places is very low in high altitude, where the malaria is not there at all. Okay? And on the other hand, if you compare where the malaria is prevalent in Chhattisgarh, for example, so there is a mutation which actually causes the sickle cell disease. That's because of the high influence of malarial parasite. The sickle cell mutation has caused sickle cell, hence the malaria parasite unable to enter the cell. So there are evolutionary force which makes some other mutation in some place. It's not making mutation other place. That's regarding the mutation. I don't know whether he's also talking about non-communicable disease or genetic disease.
Thank you, sir. Thank you. One more question. I think it is from Tamil Nadu, southern part of India. Is it true that Fulani tribal from Africa, their genome is resembling the particular Dravidian community in Tamil Nadu?
What is the name of the tribe?
The Fulani tribal of Africa.
No, I don't think so, because the divergence between Indians and even the Africans who are with a similar phenotype as Africans, genome is different because of the isolation. We acquire a lot of mutations. I don't think now there will be similarity between us.
Okay, sir. One more question posted by Lavanya. Do man in India could be oldest than Africa? Her question is, no idea. The last question. So since we have more questions from participants, we selected only a few questions. The last question of this session: why there is no mitochondrial contribution from parental side? Then evolutionary any evolutionary significance about this property?
Paternal contribution, right?
Yes, yeah, paternal side.
If you look at the sperm structure, head piece, mid piece, and the tail, the mitochondria present in the mid piece of the sperm, and that remains outside at the time of fertilization. So even if it goes there, it will degrade. So that's the reason there is no paternal contribution of mitochondria.
Thank you, sir. Thank you for answering all the questions, sir. So with this, we will come to the end. You can go.
Yes, sir. Yes, sir. Thank you, sirs. Gratitude unlocks Gratitude unlocks fullness of every kind virtue. Hence, I now invite the organizing secretary, Dr. H. David Raja, to propose the vote of thanks in formal. Please, sir.
Like, your microphone is mute.
Okay, sorry. A very pleasant good afternoon to all. It is a privilege and great joy for me to extend a vote of thanks in this grand webinar, Human Origin and Health and Diseases. First of all, I would like to thank and praise God Almighty to giving this great opportunity. Thank you, God. Next, I would like to thank Reverend Father Principal, Dr. M. Arakesami Xavier S.J. for his nice felicitation and blessings. Reverend Father Principal is a guiding and encouraging force in implementing many innovative academic activities in our college. In this great occasion, I would like to extend my sincere thanks to Reverend Father Principal. Thank you, Father. I wish to express my sincere thanks to Dr. K. Tangaraj, Chief Scientist, CSIR, CCMP Hyderabad, the resource person of today's webinar. Sir, you made this day fruitful one because your presentation is more informative and very useful for the current situation. Thank you so much for accepting our invitation to be here as a resource person. Thank you so much, sir. I also thank Dr. Sagai Satish, Dean of Biological Sciences, and Dr. S.R. Sindhil Kumar, Head of the Department of Botany, for their wonderful support and guidance. I especially thank Dr. Kanike Raj, the technical team head, and his team members for their excellent arrangement and guidance throughout the program. Thank you, sir. I convey my sincere thanks to Dr. T. Francis Xavier, one of the main organizing secretary of this webinar, for his hard work and meticulous planning. I also express my deep regards and thanks to all the staff members of Life Science Department for their fullest participation, encouragement. Thank you, sirs. I wish to express my profuse thanks to our PhD scholar and all the student volunteers for the e-invitation and certificate preparation. Thank you, friends. I am very glad to thank our webinar moderator, Mr. Benedict Matthew, illustrious alumnus of our department, for his wonderful MC. Thank you, Mr. Benedict. Now, the most important, my special and hearty thanks to all the participants from various parts of India and abroad for your active participation and response. Thank you, friends. Before concluding, let me express my sincere gratitude and appreciation to all the well-wishers of this webinar for the grand success. Once again, thank you one and all. Stay home, stay safe, have a nice day.
Thank you, Dr. David Raja. So at this juncture, on this special occasion, I would like to thank Dr. David Raja for his complete support for this one-day webinar. So I would like to inform our participants. So the feedback link posted in the chat box, it will be available for next 20 minutes. So please fill the feedback form. So I repeat, the feedback link will be posted. At least we already posted, so it is available in the chat box. So dear participants, please fill the feedback. Benedict?
Yes, sir.
Yeah, you wind up.
Thank you, sir. With this, we come to the end of our webinar session. Thank you so much for all the participation. Thank you.
Thank you, sir. Thank you so much, sir.
Thank you.
Okay, Xavier, wish you all the best.
Yes, sir. Thank you so much. Thank you, sir. Yes, sir.
Sir, thank you so much, sir. It's a very nice. Thank you, sir.
Thank you. Congratulations, Dr. Francis, and
thank you.
Thank you, sir.
Okay.
சார் காணிக்கிறேன் சார்.
சார் அவரோட இது ஆஃப்ல இருக்கு சார்.
ஓகே ஓகே, நம்ம shall we leave the
எஸ் சார் ஓகே சார்.
yeah okay, thank you.
தேங்க்
Sir, thank you so
Sir.
much for your technical support, sir. Kanigarai, sir. Thank you.
ஹலோ
Switch off பண்ணிட்டாரு. இது on off பண்ணிட்டாரு. Okay, thank you.
சார் தேங்க்யூ சார், தேங்க்யூ ஸோ மச், தேங்க்யூ.